Cheers, Yannis.
Morning, everybody. My name's Professor Richard Davison. I'm actually the chair of the Cycling Science Steering Group of the WCSS. It's my pleasure to be the chair for this session. Now, if you're like myself, who is a sports scientist but also a coach and a coach educator, then the topic of this symposium is of great interest, because we're always interested about, I suppose, what's the potential within the athlete that we've got, how much of it is actually born and how much can we manipulate and improve. So we're going to run with the order today of Mr. Colin Moran, Dr. Colin Moran, who's from the University of Stirling. We're going to kick off, followed by Professor Yannis Pitsalidis from the University of Brighton, and hopefully we'll get some answers to our question about what potential we have within the genes that we're given when we're born. Okay, thank you very much.
Okay, so thanks Richard for the introduction. So and thanks to you for packing out the room. I think we've got an exciting couple of days ahead of us. So as is kind of obvious from my title, I'm a geneticist by training who's kind of worked my way gradually into sports science, not least by taking a postdoc with Yannis when I was a little bit younger than I am now. So to tell you, as I understand the audience, and Richard's just mentioned it, we've got coaches, cyclists, some scientists, maybe some medics, maybe some journalists in the audience. So I'm going to give a bit more of the background genetics than I might normally do for this kind of talk, just to make sure that I don't lose anyone. If you know everything about genetics and I go too slowly for you, I apologise. If you know nothing about genetics and I go too fast for you, well then, clearly I've failed. Hopefully I'm going to strike a nice balance somewhere in between. But clearly, to tell you about genetics and epigenetics, I'm going to have to explain to you a little bit about what geneticists do and what those subjects are. But actually, a title that I might more normally use for this kind of talk, and something which really kind of describes my work area, would be to say that what interests me is, why are we not all the same? So in the context of what we're talking about for the next couple of days, when we jump on a bike, having never been on one before, why do some people go faster than others? If we all do exactly the same amount of training, do we all respond in the same way? No, is the answer. So if I get this completely wrong, if I pitch it at the wrong level, and you only take one thing away from what I say in the next kind of 20 minutes or so, is we are not all the same. And a large part of that can be explained by genetics. So if you're in a training club and there's somebody just a little bit ahead of you, and you think, all I need to do to beat them is just work harder than they do, that's not necessarily true. You might already be working harder than they are, and you're not keeping up with them. So don't feel quite so bad about it. So where this kind of area starts, for me, I think this is a nice place to start, is in the 1960s, very famous exercise physiologist, a lot of you may have heard of him, Per-Olof Astrand. He was asked a question at an exercise symposium, and his reply was that I am convinced that anyone interested in winning Olympic gold medals must select his or her parents very carefully. So he was recognising in the 1960s the impact of understanding genetics, or the impact it was going to have on him trying to understand the area that interested him, exercise performance. And this quote is more often reduced to the slightly pithier, choose your parents well. Clearly an impossible task. But what he was saying was, genetics is really important and we need to understand it. So, as geneticists, broadly, we're interested in something called heredity. All geneticists, whatever our topic. And what that means is we're interested in things that run in families, things that are passed from one generation to another. Those, from my point of view, those things come in two kind of types. So, environmental things, for instance, families will typically share their geography. So they will either all live at altitude, and we know there are benefits to altitude training, or they will all live at sea level. So they will share that aspect of their life. There either will be access to a velodrome nearby, or there won't be access to a velodrome nearby. So they will have shared facilities available to them. They'll also share, which is the pointer, middle one, which doesn't seem to work, not to worry. They'll also share their food. Typically, maybe not exactly, but typically, somebody will cook in the house for everyone, so they'll have the same kind of diet growing up. From my point of view, the interesting thing that they share is they share their DNA, they share their genes. And importantly, we know how much of it they share. So mum and dad hopefully share none of their DNA, they're unrelated, but they each pass half of their DNA on to their children. And as a consequence, the children also share half their DNA. So what is DNA? Or a full complement of DNA we would describe as a genome. And a genome, if you've heard of it before, you've almost certainly heard it described as the blueprint for life. It is the instructions for building each one of us. But if we look around the room, pretty quickly we see that it builds each one of us differently. We're tall, short, normally I would say fat, thin, but I don't think that's true in this audience actually. And that suggests that the instructions are building each one of us differently. So it suggests that there are differences in the instructions that are used to build us. Sorry, the blueprint. But what does DNA look like, and what might these differences look like? So anyone who has watched CSI will undoubtedly have seen a string of letters scrolling across the screen as they process their infinitesimally small sample and catch the killer, who is always in the database. But they're not totally wrong. So DNA is made up of 4 letters, A, C, T, and G, and it is incredibly long. It's 3,000 million bases long. How do we know that? Well, we know that a variety of different sources, but probably the most definitive source, if you like, is the Human Genome Project. Again, that's something you might have heard of. It was finished kind of 10, 11 years ago now. And that was started in the 80s. A group of scientists got together and they decided that they wanted to read the instructions of an entire set of instructions from one person. So they wanted to read every single letter. We call it sequencing. And they managed to finish it, as I say, 10 or 11 years ago. And they have it printed off somewhere. It's a wall full of very thick books. And clearly, you know, not very intuitive when you read it. But on one hand, you might think, well, that's it. Human genetics is done. We've got the sequence of the human genome. We know it all. But of course we don't. It's like having a book in a language that we don't really understand. Our grammar is poor and our spelling's even worse. But we're getting there. We're learning things all the time. And one of the ways that we're learning things is we didn't stop at a single genome. So we've now sequenced hundreds of species and we've sequenced hundreds of other people. And that allows us to ask 2 broad questions. We can ask, how similar are we genetically to other species? And if you compare that to how similar are we in our physiology, you can learn something about how genetics determines physiology. And we can also ask, how similar are we to each other? So we can learn about how genetics might determine inter-individual differences in our physiology. So you might be surprised to know that you share about 71-72% of your DNA with a horse. You share about 97% of your DNA with a chimpanzee. And you share about 99.7% of your DNA with each other. So that's no great surprise. We're all humans. You'd expect us to be more similar to each other than we are to other species. But 99.7%, that sounds like we're almost identical. Sounds like there's nothing terribly interesting there. 0.3% of the genome at which we vary, if you combine that with the vast size of the genome, you end up realizing that we differ at around about 10 million sites in our genome. So about 10 million of those letters, we don't all have the same letters. One person is an A, another person is a C. And where those differences occur in genes, they give us different versions of those genes. So we tend to think of these in very visual terms. If this difference occurs in the eye colour gene or the hypothetical eye colour gene, we might have a blue-eyed version and a brown-eyed version, or blonde hair or red hair, if it's in hair colour genes. But this can also be true in genes that relate to performance, as I'm going to try and convince you. So we know that there is a very large amount of genetic variation. That's clear. But if we want to understand how it relates to performance, we need to understand performance variation. So we took everybody in this room outside, right now, put them on bikes and had a race. Somebody would finish first, somebody would finish last, quite possibly me, and there would be people spread out in between. But at that stage, all we would have is a result. We wouldn't know what explains the result. Why are there differences? It could be about differences in technology. It could be about differences in training. Perhaps some people do lots of training, some people don't. Could be because we've got men and women in the room, who's going to go faster. We've got young people and old people in the room, or at least we've got Yannis. So. It could be about nutrition. Some people might have a better diet, some people might have nutritional strategies for the race, or it could be about genetics. So clearly, to answer this kind of question, we need to do carefully controlled scientific studies, and an excellent one has been done quite a number of years ago now, 15 years ago. This is known as the Heritage Study. It was done by Claude Bouchard in Quebec. And what they did was they recruited just under 500 sedentary individuals. So they took the training aspect out of it. They knew that these people didn't do exercise. They measured something called their VO2 max. It was mentioned downstairs. I imagine most people know what that is, but if you don't, it's the maximal rate at which your body can use oxygen when you're exercising. And in layman's terms, it's how much puff have you got. Higher is better, basically. So they measured VO2 max in these individuals, had entry to the study, and the average was about 2.5 litres. They then put them through 20 weeks of aerobic exercise training, and from memory I think they went to the gym 5 times a week, and they did the kind of things that most people do when they go to the gym. They got on treadmills, they got on bikes, they got on rowing machines. They were doing things that were designed to improve their VO2 max, to make them aerobically fitter. And unsurprisingly, they got fitter. The average increase in VO2 max was about 400 mils. But what was really interesting from a geneticist's point of view was the range of improvements. So what you can see up at the top here is that the biggest improvement in any individual was about 1 litre, so about a 40% improvement in their VO2 max with 20 weeks of aerobic exercise training. This is change in VO2 max on the y-axis, and this is all the individuals lined up next to each other. So you can see that over here, some people after 20 weeks of aerobic exercise training got no benefit whatsoever. No improvement in their VO2 max. They might as well not have bothered. From my point of view, what's interesting is this is not a dichotomous relationship. It's not non-responders and responders. It's everything in between. And that is a sort of classic quantitative genetics phenotype that we would hope to be able to explain with genetics. So the story doesn't end there. So Claude Bouchard had not recruited unrelated individuals. He'd recruited families. Remember I said at the beginning that we know how much of their environment they share, or we can estimate it, and we know how much of their DNA they share. And with a bit of maths that is kind of standard in this sort of genetic study, we can estimate how much of this variation, so where you fall on this curve, we can estimate how well that can be explained by genetics. And what we find is that about 50% of this variance can be explained by differences in genetics. And what was also interesting was they had corrected this data for your baseline VO2 max. So this particular aspect of genetics that explains response to training was unrelated to where you started off. And in fact, when they do this for where you start off, they found that around about 50% of that could be explained by genetics as well, but different genetics, or different genes. So, the way I always think of it is this. To become a champion, if this is 0, starting down here, and the podium is up here somewhere, you first of all need to have the right versions of certain genes to have natural talent. So we don't tend to think of that as genetics. We all talk about somebody being naturally talented, but that's their baseline genetics. That's your starting point. So to be a champion, you have to start off kind of high. But if you start off high and you've got the wrong genetics for responding to exercise, you're not going to make it. So you've also got to have the right versions of certain genes to be able to respond to training. And of course, if you've got both of those, but you spend your time sitting in front of the computer playing computer games, you're never going to be a champion or a sports champion. So you also need the right environment, and I'm grouping everything else together there. So you need to be training, you need to be doing the right kind of training. At the elite level, you need to have the right kind of support, the right technology, all the other things that we're going to hear about in the next couple of days. So that was 15 years ago, and at that stage we don't know what specific genes are involved. The way the maths works is we can estimate the size of the genetic effect without knowing the specific genes. We've been doing it now for 15 years, and most years there's a report published, usually involving Claude Bouchard and several other of the top people. But in essence, what I want to say on this slide is that we've now identified over 200 genes that are involved in how we perform exercise and how well we perform exercise. So genetics, something I meant to say in the last slide, if genetics is explaining 50% of the variance, then what that means is that it's at least as important as everything else put together. And we now have identified over 200 genes that are involved in performance. So it's really important to understand it. So for the purposes of time this morning, I'm just going to give you one example. And this was probably the first performance gene that was identified. It's a gene called ACE. The reason I chose it is because many of you might have heard of it. You may have heard of it, or you're most likely to have heard of it, in the context of ACE inhibitors. Pointer does work. No, yeah, it's a bit intermittent. So if somebody has high blood pressure, maybe somebody you know or relative, they're quite likely to have got ACE inhibitors from their doctor. So this would be the gene that they would work on. And what it normally does is it converts something called angiotensin 1 into angiotensin 2. Yannis, could you go into my bag? There's a pointer in there.
You could use the mouse cursor or something else.
That's true, actually, I could do that, don't worry about it. So it converts an inactive peptide angiotensin 1 to angiotensin 2. In turn, angiotensin 2, it's a potent vasoconstrictor, so it makes your blood vessels smaller. And that in turn increases your blood pressure. However, so ACE comes in two different versions. There's what we call the D allele, or deletion allele, and the insertion allele. And the way these differ is that the I allele is associated with lower levels of circulating ACE. So if you've got lower levels of ACE, you get less conversion of angiotensin 1 to angiotensin 2, you get less vasoconstriction, and therefore you get lower blood pressure. Or in the context of performance, what we think is happening is you get altered blood flow around the body. And there are many papers on ACE. So I've chosen a very early one. This is by Hugh Montgomery's lab. And what they did was they looked at how this genetic variant associated with running performance. They had 91 Olympic standard runners and about 2,000 controls. And what you can see is that the I allele is at around about 50% in the controls. Whereas runners who did shorter distances were less likely to have the I allele. So they're less than 200 meters, they're sprinters. These guys would be the endurance athletes, over 5,000 meters, and they've got higher levels of the I allele. And the intermediate distance group were intermediate. So you've got this nice climb, showing that the higher the carriers of the I allele are more likely to be good at endurance. Okay, so the problem with all this genetics, all these 200 genes, is that each one of them only explains a really small percentage of the variance. The biggest ones explain about 1 to 2% of the variance that we see. They don't generally, if we look at lots of different performance phenotypes, they don't generally explain the 50% of the variance that we know influences most of them. So, what could be the reason for that? Well, one possible reason is that we've tended to look at what we call common variants. So in terms of genetics, that means that there are variants that are at a frequency of greater than 1% in the population. If you think about that, a consequence of them being common means that they probably have very small effects on function, because if they didn't, evolution would select against them. So they're easily tolerated. They're also easy and cheap to analyse. So they're easy in the sense that they're common, so we don't need to collect very large numbers of people together to be able to identify both groups within our cohort to see how they compare to each other, both genotype groups. Also, we are beholden to the companies that produce the technologies that allow us to look at these, and of course they want to sell their product, so they make a product that works with the mass market. Rather than being interested in things that are rare, that a few geneticists might like, they want things that they can make lots of money off. However, we are beginning to look at rare variants now. So rare variants are variants that occur at less than 1%. These are less well tolerated. I mean, they're just the opposite of common variants in terms of what we're discussing. They may have large effects on function. Some of them will, some of them won't. It's only been feasible to analyse them probably for the last sort of 5 or so years, as we've got now what we call next generation sequencing. So do you remember at the start I talked about we sequenced more than one person and more than one species? The first genome cost off the top of my head, I think it was about $3 billion. We can now do one human genome for less than $1,000. And it took, the first one took 15 years, and now we can do it in a couple of days. So the cost has been plummeting, and that allows us to do this on a much larger scale and to look at rare variants. So we're beginning to do this, and as is the norm with these things, we begin by looking in disease populations. There's more money in medical science than there is in sports science. And in disease populations, we are seeing that some rare variants do contribute to disease. But it's not been done in athletes yet, or in exercise population. In my opinion, it will be shown that they also influence athletic performance. So as a way to back that point up, I can give you one example from the literature. Now, this was not found using next generation sequencing. It was found with a slightly more old-fashioned, lower throughput method. But this guy here, Eero Mantaranta, and if we have any Finns in the audience, I apologise for my pronunciation. I have no idea how I meant to say that. He's a very famous cross-country skier from the 1960s, and you can see he won a clatter of medals at the Olympics. 7 Olympic medals, 5 World Championship medals, 9 domestic championship medals. Eero had a really unusual advantage over his fellow competitors. EPO. I don't think this needs any introduction in a cycling room full of cycling enthusiasts. EPO, what it's normally doing is normally when you have low levels of oxygen, EPO is produced by the kidney, and it binds to its receptor, the EPO receptor, converting it from an inactive form to an active form. This leads to increased red blood cell production and therefore increased oxygen carrying capacity in the blood. I'm sure that's something that everyone knows. Eero suffered from something called familial erythrocytosis 1. This is a very rare genetic disease. And it's caused by this variant here. He's got a rare variant of his EPO receptor. And the consequence is that he's hypersensitive to EPO. So very small amounts of EPO lead to lots of active EPO receptor. So without much EPO, he gets increased blood cell production and increased oxygen carrying capacity of his blood. And in fact, people who suffer from this can have up to 50% increase in their blood's oxygen carrying capacity. Now clearly that gives them a massive advantage at endurance events. So this particular variant was so rare that I couldn't find any information on how frequent it actually was. It's so rare nobody's bothered to look at it. But what I could find was information about the disease. So if you group 4 very similar diseases together, couldn't find information just about the single disease that Eero had, but if you group 4 similar diseases together, this occurs at less than 1 in 100,000 people. So his particular variant is going to be even less than that. So clearly very rare, but a very large advantage that it was giving him. Could that explain why certain people are Olympic champions? It's going to be part of the story, I think. Are common and rare variants the whole story? I think that's unlikely. It's going to tell us something and we're going to need to understand it, but I think we're also going to have to look at something called epigenetics. So for the last bit of my talk, the last 5 minutes or so, I'm just going to shift gears slightly and tell you about epigenetics. So what we've been doing so far is we've been talking about variants in the DNA, which is up here. So a change in the promoter might lead to an increased amount of RNA, which in turn would lead to an increased amount of protein and some sort of altered function. It might be a change in the actual exon sequence here, which is also carried in the RNA and would lead to a change in the sequence of the protein. That would be the kind of thing that Eero had, a change in his receptor, leading to altered function. But actually, 90% of the human genome is transcribed, but the vast majority of it never makes it beyond this stage. The vast majority of it is we would describe as non-coding RNAs. But they seem to have, we're recognizing now, they seem to have important functions. And we're going to need to understand those as we go into the future, to understand how they contribute to performance as well. So I'm going to tell you about one specific one specific group called microRNAs. So as the name suggests, these are short non-coding RNAs, 22 nucleotides long. They're involved in something called post-transcriptional regulation. So they're made in the normal way in the nucleus like any other gene. That's your DNA being transcribed into RNA, gets processed a bit, ends up out in the cytoplasm. And out in the cytoplasm, these microRNAs bind to the 3' UTR of messenger RNA, so that's the right-hand end. And depending on how they bind, whether it's a perfect match or an imperfect match, they either cause the message to be degraded or they cause it to be stalled. So they stop it turning from messenger RNA into protein. So they're regulating what genes do. So the genes could have the same sequence or in two people, but be at different levels because of microRNAs. What's important from a genetics point of view is that each microRNA binds to multiple messenger RNAs. So they can be controlling whole pathways, not just individual genes. But also each messenger RNA is bound by multiple microRNAs. So that means that this system allows incredibly fine control over gene expression and protein translation, rather. When I got interested in this was about 4 years ago, and it was on the back of a discovery that microRNAs, although they operate inside the cell, they're also found in the plasma. So we know if somebody exercises their legs repeatedly, that doesn't just affect the muscles that they're exercising, there are whole body effects. But we don't fully understand how that's communicated from the exercising muscle to the rest of the body. MicroRNAs are one potential source of that communication. We don't know the answer yet, but they do seem, plasma microRNAs at least, seem to be involved in cell-cell communication. So they can be produced by one cell, but affect transcription and translation in multiple cell types and multiple organs. So the paper that specifically interested me in this was 3 years ago by a group called Baggish, and I meant to check this, I apologise if I get this wrong, I think they're out in the States. And what they did was they took 10 students who just joined university and they wanted to join the rowing club. They did a VO2 max test on the bike, so exercising to exhaustion. They took a blood sample before and after. They then put them through 90 days of aerobic training, specifically designed to get them into the team to allow them to compete as rowers. And then they did the same VO2 max test on the bike at the end. So the bike takes out any learned element of improved technique. And they looked at the microRNA profile in the plasma. They looked at a lot of microRNAs in these people. Here's just an example, microRNA 146A. What they show is that on the baseline test on the bike, you can see that there's an increase in the level of the microRNA. So there's an acute response to exercise. Post-exercise training on the bike, there's also an acute response to exercise. This time it's slightly bigger. But if you look at resting before training and resting after training, you can also see that this microRNA has gone up with training. So it's responding acutely and it's responding chronically. And if you look at that, the change in the resting levels, the chronic response, you can see that that correlates pretty nicely with the improvement in VO2 max that these subjects were gaining. So what we then had a cohort of samples that we wanted to use ourselves. So we've got 407 Lithuanian athletes and controls, including Olympians. We picked out 10 endurance athletes, 10 strength athletes. We've actually got 10 controls as well, although the data's not on this slide. And we measured 5 microRNAs that Baggish had shown to change with training. We actually measured about 10, but we're showing 5 in this slide. And what you can see is that in endurance athletes, 4 of them, 21, 146A, the one we've just talked about, 221 and 222, are all increased in endurance athletes compared to strength athletes. And what's not on this slide is that the controls in all cases are intermediate, suggesting that increasing them is creating an endurance phenotype and decreasing them is helping you create a strength phenotype. At least that's one interpretation. Another interpretation that might be more interesting to some of you would be that actually it's nothing to do with training. It's that the endurance athletes had higher levels of these microRNAs before they started training, and that helped them become good endurance athletes and get reward from doing the training and therefore keep at it, whereas the strength athletes, or the guys who became strength athletes, didn't have that. They were getting a reward from strength training rather than endurance. So we don't know that from the study that we've done. And that's just what I've said there. So just to summarise, my take-home message, if I lost you completely, we're not all the same, and in part that can be explained by genetics. That's the point I really want to get across to people. In part it can also be explained by epigenetics, and although I've got environment as something separate here, what I actually think is that epigenetics is probably the way that environmental stimuli and training are being encoded into this system. So in my opinion, to win the Tour de France, you must have a mix of everything. You've got to have the right genetics, the right epigenetics, training, nutrition, technology, team, and I'm sure a whole lot of other things that I've missed out. So finally, just like to, for the last study, thank my collaborators. Sophie Wardle's my PhD student who did the work in the lab, and Kevin Tipton and I are our co-supervisors. These are my collaborators in Lithuania who collected the samples, and some at Glasgow as well. And this is my university. Okay, thank you for listening.
Well, good morning, everyone. I'd like to thank the organizers for inviting me here to present to you. The program is indeed fascinating, and I'm sure we'll enjoy it the next few days. I think you'll also agree that Colin has beautifully summarized the area and convinced us, if anyone wasn't convinced, of the importance of genetics. What I'm going to try and do is try and share with you, well, how do we find which are those genes? And what achievements have been made in all these years. In other words, how can we apply some of this knowledge? And from the point of view of the organizers, I've only got 14 minutes left. There's no way I can do it in 40 minutes. So hopefully I won't keep you too long. Okay, so I think Colin also has said quite a few of the things that I'm going to say. So, for time, I will go over some things quite fast, the things that Colin's already mentioned. And he has already referred to the notion that elite performance, or in this case world-class performance, is going to be made up of a number of factors. I'd like to use this three-legged stool illustration of the importance of genetics, training, preparation, and clearly genetics is going to have a role, as we also heard from Colin, in training and even in preparation. And for a world-class performance like Usain Bolt's performances, if one of those legs were to break, you'll have disaster, lack of performance, and even the great Bolt loses races. Okay, so what we really need for a world-class performance is to get all these things right. But also, as Colin mentioned, the famous quote by Professor Astrand is choosing your parents important, and I certainly believe this one here is of paramount importance. You can develop this, you can prepare well, but if you don't have this, you have a major problem. And this importance of genetics, going back to some historical information, is nicely illustrated going back to my Greek hereditary, which is Hippocrates here, who said this whole notion of positive health, and I'll read this to you. He said that positive health requires a knowledge of man's primary constitution, and by that, this was what he meant at the time, obviously, is genetics, and of the powers of various foods, both those natural to them and those resulting from human skill. But eating alone is not enough for health. There must also be exercise, of which the effects must likewise be known. The combination of these two things make regimen. When proper attention is given to the season of the year, the changes of the wind, the age of the individual, and the situation of his home, if there's any deficiency in food or exercise, you can't see that, it says the person will fall ill. And I think what we're going to find out, or what will be confirmed in the next 2 or 3 days, is very much what Hippocrates said, think back 460 to 370 BC. Do we really know much more than this? Well, if we go a bit further along, and we go now to 1869, I could refer to Charles Darwin's work, but you know that quite well, and I'd rather refer to his cousin, Francis Galton, who wrote this book, which I very much encourage you to read if you have time. I have the web address here if you want to download it. And it was called Hereditary Genius, Its Laws and Consequences. And in case you don't have time to read it, I'll just focus on one of his chapters, which again I think illustrates some of the points I want to get across. In this chapter, classification of men according to their natural gifts, he says, everybody who has trained himself to physical exercises discovers the extent of his muscular powers to a nicety. When he begins to walk, to row, to use the dumbbells, or to run, he finds, to his great delight, that his thews strengthen and his endurance of fatigue increases day after day. So long as he is a novice, he perhaps flatters himself, there's hardly an assignable limit to the education of his muscles. But the daily gain is soon discovered to diminish, and at last it vanishes altogether. His maximum performance becomes a rigidly determined quantity. He learns to an inch how high or how far he can jump when he has attained the highest state of training. He learns to half a pound the force he can exert on the dynamometer by compressing it. He can strike a blow against the machine used to measure impact and drive its index to a certain graduation, but no further. So it is in the running, in rowing, in walking, and every other form of physical exertion. There's a definite limit to the muscular powers of every man, which he cannot by any education or exertion overpass. We heard early on this morning Ross Tucker talking about breaking these barriers. Well, Galton told us this some hundreds of years ago. And he goes on in this book to talk about the importance of genetics in mental performance and in particular IQ, and you can read that yourself. The importance of genetics, I've mentioned here Galton, Hippocrates and so on, but a lot of what the public read is influenced by writers and popular writers, and I'm sure probably most of you, if not all of you, have read at least 3 of these 4 books. Malcolm Gladwell's Outliers, David Shank's the Genius in All of Us, and certainly Matthew Syed's Bounce. Here they argue very much of the genetic talent myth and the power of practice. The genes are not really that important. And I've written one book that no one has read, but these individuals, okay, are making a lot of money by publishing their books, and those are the messages that get across into the public. And the notion is that everyone can. Well, I'm afraid not everyone can. These books are very much influenced by the work of Anders Ericsson, and again, I'm pretty sure you know his work quite well. I'll just remind you of some of the important bits published back in 1993. He wrote, many characteristics once believed to reflect innate talent are actually the result of intense practice extended for a minimum of 10 years. The whole notion that 10 years required to produce expert performance. And this message has really very much influenced a lot of the sporting bodies. But what is the science behind that? And he also writes, and I'll highlight this important bit, practice is both necessary and sufficient for the attainment of deliberate performance. And it's effective because it selectively activates dormant genes that are contained within all healthy individuals' DNA. Again, in other words, all of us can achieve it. I'm sorry, it's not like that. And there's absolutely no evidence for this whatsoever. And going back in 2012, I was very fortunate to have an ACSM symposium on the topic, accepted and supported by the Physiological Society. To be fair to Anders, he did come to defend himself. Well, none of the writers of the popular books accepted my invitation to come and defend themselves in a scientific audience. That tells you a lot. So I have a lot of respect for Anders coming to defend himself and really defending himself in front of the real big names in sports genetics, and we've already heard of Claude Bouchard's work. And you can see from the title of the symposium, The Genetic Talent Myth and the 10,000-Hour Rule. I don't know if any one of you was at the symposium, but I think it would be fair for me to change the title by the end to Genetic Talent and the Myth of the 10,000-Hour Rule. And I think that is really the correct way of putting this. But I think that symposium and the dinner we had that evening probably did influence Anders' work, because the same year he published this paper, and I'll just read what he wrote here. With the recent advances in genome-wide mapping studies and the emerging findings on the relation between athletes' training histories and their performance, this should be a time for integrating these two bodies of knowledge for a more complete understanding of the complex development of elite performance. Here for the first time he's telling us actually genes are important, and I think we probably convinced him of that, and Colin has summarized some of that literature. And he was actually responding in that paper to some of Ross Tucker's work together with Malcolm Collins, What Makes Champions, a review of the relevant contribution of genes in training. And this is a different way of saying some of the things that Colin's already mentioned. And this is taking 6 theoretical individuals. Here we have performance going across to the current elite performance threshold and the current world record. Here we have performance level at the first exposure, and this is the maximum performance range. Here we have 2 individuals who have this innate ability, but they don't know that. They haven't been trained. And then we have 4 other individuals here who, with training, have reached this point. This is where they could theoretically reach if they had all the proper training. This individual here can attain this level. This person here, probably myself, with all the training in the world, would not make it over here in the sport that they wished, because I didn't choose my parents well for volleyball, which was my sport at the time as a young Greek. But the point is, well, how do you find the genes that are important, that influence this, so we can actually find this individual in our talent ID programs? And that is really a quest that I've been on for 15 years, and I'm afraid that I'm going to disappoint you by the end of today. We know very little, but we're making some progress, and I'll share with you that progress and what we need to do to really make progress. But some companies will tell you, we already know this, they've already achieved this, and this is a slide that I took, or a photograph that was taken when I was boarding a flight from Tallinn airport to fly to Barcelona for the ECSS conference a few years ago. And right next to my gate, you could actually provide a DNA sample, and by the time you land on the other side, they'll tell you what sport you designed for. And you can see this procedure involves taking a DNA sample, scrubbing inside your cheeks, blah, blah, blah. You get the results in 14 days. Test costs 95 euros. I can go through other companies that can tell you they'll do it for 1,000 euros and give you every single thing you'd want, in addition of how not to get old. Okay? And the problem is the public believe these kind of messages because we have failed as scientists. No one reads our books, no one reads our papers. So another way of presenting, again, what Colin mentioned, this notion of the environmental factors and individual genes being important to elite performance. How do you find these genes? And that's really what I want to focus on, and the progress we have made. And I'm going to use examples from athletics. That's where we've made most of our progress. And it was important to find enough individuals to do our experiments. Typically in genetics of health, the cohort size of over 100,000. Trying to find 100,000 Olympians in a sport, you can imagine, are very difficult, especially you're trying to also include and take into account ethnicity and so on. So this is the medal tally from the Moscow World Championships of last year. The countries that you really want to do your research in genetics are Russia, United States, Jamaica, Kenya, Germany, Ethiopia, and Great Britain. These are the ones you want. These are the top 7 countries. And for today's purpose, I'm going to really focus very much on Kenya and Ethiopia, and maybe a little bit on Russia as well, but we'll come back to that. And I'll show you why I'm selecting those countries. Now, this is the World Championships results for 800 meters to the marathon in the men, 800 to the marathon in the women, the gold, silver, and bronze. Now, watch when I put the results up. That is Kenya. That is Ethiopia, the East African region. It doesn't end there if this doesn't impress you already. If we put the athlete from Djibouti, we put Mo Farah, allow me to say he's from Somalia, and then we have the athlete from Uganda, East African region. You'll see that almost every gold medal was won by an East African, with the exception of one. What is this telling us? Well, one would argue that this is telling us that genetics are important, but from my point of view, those are the cohorts I want to look at to find the genes that are important. And that impressive fact doesn't end there. If you look at the world marathon record, we have Paul Tergat under 2.5, Haile Gebrselassie under 2.4, another Kenyan here, Macau, going to 2.338, and the current world record holder 2.323. Again, all from the same region. These guys must have the right genes, given what we've heard as well from Colin. So, what makes these athletes great? Is it genes or is it environment? Now, I want you to actually choose one or the other. How many of you say genes? Put your hand up, right high up so I can see. Okay? Alright, most of you are saying that it's genes. Okay? So you're saying, in other words, that these populations, the East Africans, have superior genetics to the rest of us. Okay? That's what you're saying. Well, I will disagree with you, and I'll by the end of my talk, and I'll try and explain to you why I disagree with you. And going back to 2004, we set up the International Center of East African Running Science, called the ICEERS. Actually, Colin Moran was the person who coined the term of ICEERS. And at that time, 2004, we brought together all the experts to explain why these East Africans do so well. And we have a book which I said no one has read. But one of the big aspects was the superior genetics. So now it's 10 years since we set up ICEERS. What have we found as far as supporting that notion, given that you're also saying it must be genetics? And these are some of the other factors I don't have time to go over. Starting the sport from a very young age, role models, how's your training, very hard training, bone breaking, and the diet. And these are some of the things that we'll hear over the next 2 days. And here's a very young Colin here who did some of the early work. And I'm just going to show you what he did, or one of the projects that he worked on together with Robert Scott. And they looked at mitochondrial and Y chromosome genetics. The nice thing about that approach is that it tells you a little bit about your ancestry, and in one sense as well, how homogeneous a population is in terms of genetics. This is the mitochondrial tree, and if you won't be able to read these numbers, but if you have those types, you're typically African. If you have those types, you're Asian. And if you have those types, you're typically European. And the question was, in Ethiopia and in Kenya, would they be predominantly of the African type, and therefore supporting what you're saying that they must have superior genetics? And that's what we wanted to do, and that's what Colin did. This is a simplified version of that mitochondrial tree. Here you have those African types, and here the Eurasian types. In green we have Ethiopian controls, and in yellow we have Ethiopian athletes. What you'll see is they're distributed all over the mitochondrial tree. They're not a homogeneous gene pool in terms of this marker. We did the same thing in Kenya. I'm not going to go over this too much time because I don't have much time. Basically the same thing, distributed all over the mitochondrial tree. Some differences here, but they did not explain the phenomenon. If I had more time I'd present you the Jamaican data where from the top Jamaican sprinters versus the controls, we find exactly the same thing. Not a homogeneous gene pool as has been suggested in the lay press. And so the conclusions from those kind of studies were that the East African athletes are not a distinct population. Some haplogroups were associated with performance but did not explain the phenomenon. And this is very important, not supportive of the idea that they arise from a limited genetic isolate. And really, since Colin left us in Glasgow, we haven't really found out much more than that. But I will share with some of the newer things that are coming out just now. Colin already mentioned this notion of the gene map and trying to find polymorphisms that may be important. And he also referred to some of the newer gene maps that have come out. And he referred to the ACE gene, which is one of the genes that's been considered important. I'm also going to refer to another gene in chromosome 11, the SPE gene, or ACTN3. And one thing that Colin didn't mention is that of the more than 200 genes that have been associated, I would argue almost all of them are false positives. Actually, not really important. And down to the fact they're small sample sizes, and if you're doing so many tests, etc., you will probably find some of these findings. But these two are probably going to be two of the important ones. Again, for the sake because of time, and since Colin has mentioned this, this just confirms what Colin already mentioned, the importance of the ACE gene, D for strength and I for endurance. And I just want to compare the East Africans to the best sprinters in the world from Jamaica. What you'll note is that these are three different genotypes. You'll note that the Kenyans are not different to the control population. The same in Ethiopia, so no difference. But important, if you compare the top marathon runners in the world to the top sprinters in the world, what would you see? They look exactly the same. So you can't have two extremes of physiology having the same genotype. That tells me this gene is not important. Go on to the sprint gene, which is probably, there's more evidence that this gene is probably the most important of all the genes that have been, let's say, alluded to be important. She wrote a few years ago, and I've taken a slide from one of her talks she gave in Glasgow, the presence of alpha-actinin 3 provides an advantage for sprint athletes. Not having this may provide advantage for the endurance athletes, so not having the version that produces the protein. And effects, there may be gender effects as well, and also going to as high a level as possible, looking at Olympians and so on. And those of you who are interested in this will have seen this data before. And this is where she got the story. And this is from looking at data or results from the Australian Institute of Sport. Here you have the control population of the 3 different genotypes. XX is the version where you don't produce the protein. So you can see about roughly 20% don't produce the protein. They'll be more suited for endurance, while the others will be more suited for the sprint events. If you note here of the sprinters, the elite sprinters, there's no one here that's got the wrong type. And in all my cohorts, they're all the elite sprinters from Jamaica, we find exactly the same thing. So this gene clearly is going to be important. Interestingly, on the endurance side, yes, something may be going on here, overrepresented compared to the controls. And there's some debate on the importance of not having this version, not having the protein version with endurance. But again, if you look at our data, I'm just going to home into the athletes from Kenya. No difference between athletes and controls. They are the 3 different genotypes. Compare the top marathon runners in the world to the top sprinters in the world. They look again exactly the same. So of 0 predictive capacity, this is really problematic as far as giving us any useful information for the practitioner in the audience to take home. And the conclusion, and I can carry on looking at other genes that we've studied, and this is illustrated in this chapter we published in this book with the IOC, that genetic studies of elite distance runners from Kenya and Ethiopia and elite sprints from Jamaica, USA and Nigeria do not find that these athletes possess a unique genetic makeup. Rather, they serve to highlight the high degree of genetic diversity among ethnic groups. It's unjustified, though, to regard ethnic difference in sporting success as genetically mediated. To justify so, one must identify the genes that are important, which until now is proven elusive. We haven't found these genes. So what progress have we made? Well, I'll come back to the kind of environmental factors that are clearly important. I don't have time to go through those. But there is a very interesting experiment that has been conducted in Kenya. Not really an experiment, but for today's purposes, since we had a cycling conference, I think you'll be very interested to see something that's going on in the same region that has produced the success of the East Africans, in particular in Kenya, where all of these things, or most of these things exist, not necessarily the role models. And I hope this video plays and you'll find this interesting. And this is the East African cycling experiment. I hope this plays. Oops. Oops. Now this is the same region where all the top Kenyan athletes are from, the Rift Valley. That's Chipkolel Stadium.
Kenyans winning marathons everywhere in the world. It ought to be really good if you gave them a bike. It was just like a thought, and I waited for the Singapore Marathon. I looked for the winners, and I said I'll just follow you back to Kenya. And I followed them all the way back, and I lined up with the Kenyan. That's how the whole project started.
Find your rhythm first. Windy, you're going to get pushed around, so find your rhythm, build into it, and then go fast.
If Nicholas is open enough and is willing to learn, and his coaches and backup team develop a very good understanding of how Kenyans think and how they approach issues, what motivates them, then it's too big stuff.
He's Radisha's coach, amongst others.
And we have to invent the ingredients. When we actually invent our own ingredients, it might even be a better cake than the one that's traditional and the recipe works. That's the premise behind it. So if we can jag this, we might have a better team than anybody else in the world.
Because of time, I'll just bypass that. Now, basically, this is the founder of this organization. He went out to Kenya. Obviously, was impressed, as we all are, the success in endurance. They allegedly have the superior genes, as you indicated. Then, if we give them a bike, they can produce the next Tour de France winning team. That was what his ambition was some 5 years ago. He hasn't achieved that yet. He believes he will, and he keeps on putting more money into it. I would argue, however, that, and I've met with him a number of times, is that the problem is that the phenomenon that we see in East Africa is not genetic. it is the environmental in a big way, as illustrating these key messages of what we've learned over the years by studying the East Africans. So I think his premise of producing a Tour de France winning team from Kenya is not going to materialize. But I do, if you do have time, do link up to that video and look at the documentary. It's a very interesting program. So what progress have we made? I'll try and do this in the next 5 minutes of what we have found more recently. So we heard from Colin. I introduced a little bit about using the candidate gene approach. But if there are some 22 or whatever thousand of protein coding genes and we're doing 1 or 2 a week, I'm going to run out of years. So we need to find another way. And that approach is already what's been alluded to by Colin, which is looking at the whole genome. He mentioned whole genome sequencing, which is not what we've really attempted yet. But we have very much gone along the lines of looking at a whole genome approach using the genome-wide association studies. And the notion behind this approach is that most of the data published to date in those kind of gene maps are basically, I believe, false positives, down to the fact that small numbers have been used. And the notion is if we can use athletes of higher caliber, Olympians, world record holders, maybe we can circumvent this need of having large numbers and find those elusive genes. So we need to get very good cohorts. So for today, I'm going to say, as I said, I'll focus on athletics, but we have swimming cohorts, a rugby cohort, and I hope soon a UCI cohort. We already have some of the top Spanish cyclists in our biobank, but we don't have enough of them to really do genome-wide association yet. The approach, as we already heard, is using a chip of some sort, whether it's Illumina technology or it's a Affymetrix technology. We have used Illumina only because it's a little bit cheaper. And in our experiments, and this shows you what experiments we've done so far, we've used either this chip on the left or this chip on the right. Now this chip on the left here, you're looking at over 1 million genetic variants per experiment rather than 1 or 2 genes. If you use the chip on the right, you're looking at 730,000 genes in one experiment. And you can see the cohorts that we have actually started doing. As we also heard, there's not a lot of money in this, so doing this is expensive and it's taking time. So what have we found so far? Well, in collaboration with Claude Bouchard, Alessandro Lucia, Nori Yuki, Ildis Akhmetov and Kathy North, we've created the GAMES consortium, where the discovery phase, the first level where you do this whole genome approach, we've looked at the gene athlete study, which includes athletes from Germany, Finland, Canada, and USA, as well as a Japanese cohort. We've looked at the whole genome. They tells you the numbers. And then we've gone on to replicate what genes came out to be important in the Spanish study, in samples from other European countries and Australia, and also Russia, Ethiopia, Kenya. You can see the numbers here, much greater than normally of 30, 50, 100 individuals. We've got 1,500 here. 792 and controls over 2,000. This number is almost expanding. At ACSM only a few weeks ago, Claude Bouchard and Turma Rankin from his group presented an update from this. I'm not, because of time, I'm not going to go into the details. Basically, nothing came out. Nothing strong. After all this effort, all this money, we found very little. But work still needs to progress. Maybe something comes out, and watch out for this. However, a more positive story came out of the sprinting side of the same story. And this is the way you do this genome studies and the way the data looks. This is the 22 autosomes. Here we have a p-value. And really what you want to do is you want to get a dot above this blue line or above this red line, which is a threshold we use for saying that gene or that version of the gene is important. And this is for the 88 Jamaican sprint athletes, the best in the world, the likes of Usain Bolt, versus 87 Jamaican controls. And you'll note 17 genes went above this line. Okay, 5 times 10 to the power minus 5. Interesting, are these genes important? We did the same thing in US sprinters. These are some of the best US sprinters in the world from the United States. Again, you can see the number of genes we're looking at. Now we find 7 genes that are coming above this line. Interesting, are these the elusive genes? We looked at Japanese sprinters versus controls, over 500,000 genes. Now we have 36 genes that are coming out to be important. The question we all need before we go out and produce our own company to make a lot of money over there, we found the next great, the genes of importance, is what happens when you do the meta-analysis incorporating all 3 cohorts. Interestingly, 2 genes come out to be replicated. The first one is the gene called GALANT13, And you can see the p-value in the Jamaican, African American, and Japanese being significant. I won't go over why we're confident this is probably real, because I will bore you in detail. You don't need to know that. And the second gene is the CREM gene, where again we find significance in the Japanese and the Jamaicans. And when you look at the combined p-value, it's 10 to the power of -6. And the previous slide I didn't show you, it was 10 to the power of -7. And that, from a geneticist point of view, is really quite interesting. What about these two genes, which may be the first important genes, is that there's not much data about these genes, but it's been associated with longevity, sudden cardiac arrest, childhood obesity, the CREM gene with Crohn's disease and inflammatory bowel disease. What does that mean? I don't really know. But when we were in ACSM, Ilis Akhmetov had access to this Russian cohort, very large cohort, and a great genetic capacity to generate data quickly. I asked him, please go and genotype your sprint cohorts and see what we find. Could we replicate? Well, he replicated. And I'm sorry the presentation was not very pretty, but this is what he sent me. And I'll take you right down. This is the group, is the strength group, the power group, the sprint group. And this is the power group and sprinters combined versus controls. And you can see here the G allele, 18% in the power group versus 11% in the control. It replicates. I'm not going to go through the details because of time, but that's for the Gallant-13 gene. We need to do further replication, but that is another cohort that's replicating this particular gene. What about the CREM gene? Exactly the same thing. It replicates again. Those of you who got a very good eye would note that the control group is only 95 versus the power group, which is 155. Since sending me this slide, he's gone on to increase that number to over 500. A very good control group. This p-value becomes even more significant. So what we're currently doing now, we need to replicate this further. We've asked Kathy North at the Australian Institute of Sport to replicate, with the DNA that she has from the Australian Institute of Sport, to see if she can replicate in the Australian study, and that is where the story ends. So I can see as well that Louis is coming, so I need to end, and I will end. And we published this version of what I'm trying to tell you I'll be going very, very quickly because of time, what little we know. And I think and you can read this in your own time if you want, but really the main point is that current genetic testing has zero predictive power. Talentification should not be used by athletes or coaches. So currently there's nothing useful for you to use. I've indicated what kind of progress needs to be done in order to have useful tools that we can use. And I think Colin made the point of gene expression and other types of omics that may become more useful for you to use from a practical point of view than genetics, for reasons I've alluded to. There is a bright future, I think, for the future for using this kind of technology, but it's going to take time before we can have something that will be of use to the practitioner. And the final point to make is that you're here for cycling. I've not presented any cycling genetics, because the notion is that if we want to develop these cohorts, we need to work with you who have access to the athletes to develop these kind of biobanking and replication studies that are needed, and hopefully that we can do that together at some point. And I'd like to use it to stop here because of time, and I hope I have shared something interesting with you. Thank you very much.